Very warm greeting to my listener. I am Dr. Sumiran Srivastava, Assistant Professor, Department of Biochemistry, Bundelkhand University, Jhansi. I am going to discuss on the topic of basic of molecular biology. So, before starting my presentation, I would just like to introduce about the cell. A cell is the smallest structural unit of an organism that is capable of independent function. All the biomolecule, that is the DNA, RNA, proteins, is present inside the cell. So, all life depend on three critical molecules, that is the DNA, RNA and the protein. So, DNA is hold information on how cell works, and RNA is molecule that is transcribed by the DNA molecule and act to transfer short piece of information to different points of the cell. The next one is a protein. It is a very important biomolecules. It form the enzyme and send the signal to other cells to regulate the activity. So, how these DNA, RNA and protein are synthesized? So, here is some central dogma which explain the process. First one is the replication. DNA form its copy by the process called the replication, and this DNA transcribe to RNA by the process of transcription. So, DNA can replicate and produce its copy. This is called the replication. So, information coded in the sequence of the base in DNA is passed to the molecule of RNA. This process is called the transcription, and the RNA is the molecule transcribed it processed to protein by the process called translation. So, information in RNA is passed to protein, it never passed from protein to nucleic acid. So, here DNA pass its information to RNA, and many times it also happened that this RNA molecule reverse transcribed to form the DNA, that is called the C dependent process. So, here the DNA, RNA, how they flow its information. So, where its process takes place. If you are taking about the overview of the DNA to protein to RNA, so in the nucleus, within the nucleus, nucleolus is present. So, this nucleolus consists the rRNA molecules. So, transcription occur in nucleus, and after transcription RNA is produced. This RNA is responsible for the formation of the protein by the reaction of the translation process. So, in nucleus rRNA is synthesized. This RNA transformed to cytoplasm, by which various types of transporter involved, and this translation process various types of protein form in the polypeptide form. So, if you are talking about the general about the DNA replication, in which there is the DNA form its copy, and the via DNA from the RNA, that is process is called transcription process, and this the information coded into RNA transformed to the protein. So, many times it also happen it transform to DNA from the cDNA copy, the process known as the reverse transcription. DNA is the very important, it is responsible for the code of life, because it have the double stranded structure in which the daughter strand are formed, where it is the parental strand and then another one is the daughter strand, which is formed after the replication process. So, DNA having basic 4 molecules: adenine, guanine, thymine and cytosine, which pair with adenine pair with thymine and cytosine pair with guanine form the double stranded structure. DNA is found in the very superstructure form. When we see in the metaphase stage, the DNA is highly condensed via the various types of organization, that we can say that the scaffold association of chromosome, and next one is the chromosome scaffold, in which it changes its organization and it fold the beads on a string, that is called the nucleosome model, in which histone protein involved in the nucleosome structure. So, if the replication takes place, this DNA should be open. So, DNA can replicate by the splitting of the hydrogen bond, in which both strands are separated, in which DNA polymerase associate and help to form the replication copy of the parental strand. So, by which 5 prime to 3 prime end, there is the DNA having the polarity in which the 5 prime and 3 prime end, in which the DNA polymerase associate and synthesize the daughter strand by the involving of many types of other biomolecules and enzyme called the DNA polymerase, first, second, third, DNA ligase, helicase, and the 2 strand of form. that is called the daughter strand. So, here the replication is formed in which new strand and old strand is associated, and finally the synthesis of the daughter strand, in which one parental strand synthesize the two daughter strand, in which the hydrogen bond and phosphodiester bonds are present. Next biomolecules are RNA. RNA is the similar to the DNA chemically, but it usually only having the single strand, and thymine is replaced by the uracil. Some form of RNA can form secondary structure by pairing up with itself. DNA and RNA can pair with each other. So, if you are talking about the structure of DNA, structure of the DNA and RNA, RNA is little bit different from the RNA in the structural form. It having the single stranded, but many times it pair itself and forming some loop and stem. So, this stem loop structure is the secondary structure of the RNA, in which the D loop, T-si loop and anticodon loop are present. So, how it is formed? It is formed from the DNA by the transcription process. The process of making RNA from DNA is called the transcription process and catalyzed by the transcriptase enzyme, that is the RNA polymerase enzyme, and needs a promoter region to begin the transcription. Like the replication process is started by the help of the origin sequence, like the origin sequence, transcription process is started by involving the promoter sequence. So, DNA get transcribed by the protein known as the RNA polymerase. This process builds a chain of bases that will become the mRNA. So, RNA and DNA are the similar, except the presence of the one base, that is a uracil. So, here it is the structure of the RNA and DNA. You can say it is a single stranded, and DNA is the double stranded. So, by the transcription process, maturation RNA takes place. Transcription occur in nucleus in eukaryotes and in the cytosol in the prokaryotes. The most important factor which involved in the transcription process is the sigma factor. So, sigma factor from RNA polymerase reads the promoter sequence and open a small portion of double helix in which the transcription is start. Various types of RNA polymerase involved, type 1, type 2 and type 3. So, RNA polymerase second is mainly responsible for the formation of mRNA. So, it catalyzes and formation of phosphodiester bond between the two nucleotides that link nucleotides together to form a linear chain from 5' to 3' direction and unwind the helix just ahead of the active site of the polymerization of the complementary base pairs. So, the hydrolysis of the high energy bonds of substrate nucleoside triphosphate to provide energy to drive the reaction. During the transcription process, DNA helix reform and the RNA is formed. In the transcription process, mRNA, tRNA, mRNA, all the types of RNA synthesized by the transcription process. So, when the terminator sequence is met, the polymerase halt and the release of both DNA and RNA. Splicing is the very important process in case of the eukaryotes. It takes place by the help of the spliceosome that brings together on the hnRNA and snRNP molecules. It's a variety of molecule that binds to the pre-mRNA and form the mature RNA. Here you are saying that the exons and intron both are present in eukaryotes, but exons should be removed by the process of splicing. Splicing is a very important process in which only the expressing element are present. So, 2 to 5 phosphodiester bond form between an introns, adenine residue, and the introns are removed. So, free hydroxyl group of 5' exons displace the 3' end of the intron, forming a phosphodiester bond with the 5' terminal, in which only the transcribe label, which we can say the element that is that going to the translation process here the present, and all the non-coding region are absent. All introns are removed and exons are present by the splicing process. Splicing is a very important process. In eukaryote cell, RNA is processed between transcription and translation. This complicated relationship between a DNA and protein it codes. Sometimes alternate RNA processing can lead to the alternate protein, in which our immune system affect. So, splicing is only occur in eukaryotes, is never occur in prokaryotes. Why? Because in prokaryotes totally the intron sequence are absent. So, these intron sequence are mainly the non-coding sequence, that is the in the splicing process. Absent the splicing process, another the process that is the we can say capping and tailing also takes place in the transcription process. So, if you are talking about splicing, unprocessed RNA is composed, that is called the intron and exon. At that situation, in the gene, intron and exon both are present, but introns are the non-coding element and exons are the coding element. So, for the translation step, we need only expressing sequence. So, basically exons are expressing sequence. So, introns are removed before the rest of an express element are present, which is involving in the translation process. So, after the transcription, RNA splicing takes place, in which only the exons are present, which is gone for the translation process. So, a typical eukaryotes gene having 4 to 20 introns, locating them by the analytical means is not easy. So, if you are talking about the anticodon and codon sequence, terminology used for the tRNA and the protein, that is the anticodon. We know very well the structure of tRNA, which having the various types of loops and arm. So, the most important arm is the anticodon arm, in which anticodons are present. The anticodon hydrogen bond with the codon that is present in the mRNA. So, this mRNA tRNA interact by the codon and anticodon elements. So, this anticodon element having the 3 codons which interact with the codon. So, by which the tRNA working as the adapter molecule which brings amino acid in the process of translation step. So, the sequence of 3 nucleotides in tRNA that recognize an mRNA codon through the complementary base pairing, basically the anticodon codon or interact and the translation step involved. So, in the translation process, protein is formed. Protein we know very well, that is the RNA DNA having the polarity like the 5 prime to 3 prime, and like this protein having also the polarity, that is called that C terminal and N terminal. C represent the COH free group and the N terminal represent the end of protein, in which there is the amino groups are present. So, C terminal and N terminal having the polarity of the protein, which the translation process the protein are synthesized. This protein many times go to post translation step. So, if you are talking about the general about the central dogma, what is the central dogma? We have discussed in the replication, transcription, translation and the reverse transcription, in which we have studied about the one more point, that is the reverse transcription, in which the cDNA is formed. Difference between the cDNA and gDNA, that is the genomic DNA, and cDNA is the complementary DNA. So, for the in the like the expression, if you are talking about the expression, checking the expression, we only use the cDNA, that is the complementary DNA, which is synthesized from the mRNA after the reverse transcription process. So, this cDNA is used in the expression for the checking of various types of disease in the PCR, in the real time PCR. So, cDNA is very important. This cDNA is synthesized by the reverse transcription process and the help of the reverse transcriptase enzyme. So, cDNA and gDNA, difference between the cDNA and gDNA, because the central dogma having both types of DNA, cDNA and gDNA. This genomic DNA having intron and exon both, but cDNA, which is useful for the real time process or any other application, having only exons, which is having the expressing element. So, it is synthesized by the central dogma process. So, replication, transcription, translation, that is having the central dogma process, and this central dogma is the basic of molecular biology. All the replication, transcription, translation process occur under the central dogma process. So, for the discussing the central dogma on these slides, I have referenced these types of some sites and books, which is the most important book is the Lehninger, that is the Principle of Biochemistry, and the next one is the Lodis, that is the Molecular Biology books, that is the 5th edition, and another one is the handbook is the Gene 8, Gene 7, Gene 8, both has the very nice book. These are some site in which I have used to save the image and download some image. So, these are some site from where I have used to use for the downloading the these some important image. So, now I winding up my topic, that is the basic of molecular biology. Thank you for attention. Thank you.